As we have already seen, humoral immunity is mediated by B lymphocytes and the antibodies they secrete. In this topic, we will explore cellular immunity, which is mediated by T lymphocytes. In cellular immunity, certain T cells, called cytotoxic T cells, act like foot soldiers and engage in direct cell-to-cell attacks that destroy the body's own cells when they have become infected or cancerous. Other T cells, called helper T cells, act to orchestrate the immune system's various responses. Your goals for learning are to define cytokines and list some of their functions, to list the types and subtypes of T lymphocytes, to distinguish between Class I MHC proteins and Class II MHC proteins in terms of cellular expression and the source of antigen they display, to describe T cell activation, to describe how cytotoxic T cells kill their target cells, to explain the central role of helper T cells in adaptive immunity, to describe the role of regulatory T cells. Here's what you need to know. The key characteristics of adaptive defense mechanisms. The mechanisms of innate internal host defenses, including phagocytosis, and the role of natural killer cells. The common features of B and T lymphocytes. To see definitions of terms, click the bold red words. Before exploring T cells in detail, let's consider a set of proteins, called cytokines, which play a critical role in the function of the immune system. Like the page boys of our medieval castle who run messages back and forth, cytokines transfer information within the immune system. We have already discussed one group of cytokines, the alpha and beta interferons. Click each image to discover some of the actions of cytokines.
Ouch.
Click the apoptotic cell to continue. Cytokines are a diverse set of chemical messengers that usually act locally, sometimes on the cell that secretes them, more often on cells nearby, and only occasionally via the bloodstream on cells far away. Let's look at two specific examples of cytokines that are important for T cells, interleukin-1 and interleukin-2. Recall that macrophages are part of the innate defenses. The innate defenses often mobilize the adaptive defenses by secreting cytokines, particularly interleukin-1. Interleukin-1 acts as a chemical alarm to alert the immune system to the presence of a pathogen. Click the macrophage to learn more about interleukin-1. Among interleukin-1's many actions, we will discuss three in particular. First, it mobilizes adaptive defenses by promoting activation of lymphocytes. Click the lymphocyte to activate it. Second, at higher concentrations, it and other cytokines acts as a pyrogen, that is, it resets the body's thermostat in the hypothalamus, causing fever. Click the brain to see pyrogens act. Third, it stimulates certain T cells, called helper T cells, to release interleukin-2. Click the helper T cell to release interleukin-2. As you just saw, the interleukin-2 released by helper T cells causes proliferation of activated lymphocytes, called clonal expansion. Interleukin-2 acts on the T cell that released it, as well as on nearby activated lymphocytes. The lymphocytes that mediate cellular immunity can be divided into two major classes based on the presence of certain cell surface proteins called the CD proteins. One class of lymphocytes has CD4 and the other class has CD8 proteins. Click each T cell to see CD4 and CD8 surface proteins. About 1/3 of all T cells have CD8 markers. The other 2/3 have CD4 markers. Click the most common CD marker. Just as activated B cells become plasma cells, the members of each class of T cell are destined to become different types of effectors when they are activated. Activated CD8 cells become cytotoxic T cells, the foot soldiers of the immune system that kill other cells that have been invaded or become cancerous. Activated CD4 cells usually become helper T cells, which orchestrate the immune response like an officer directing his troops. Occasionally, CD4 cells become regulatory T cells. Click the cell that will become a cytotoxic T cell. As you will see later in this topic, both CD4 and CD8 are co-receptors that bind to different kinds of MHC proteins. This binding is required to activate the T cell. CD8 binds to the type of MHC protein called Class I MHC protein, and CD4 binds to Class II MHC protein. Click the CD protein that binds to Class II MHC proteins. The human immunodeficiency virus, HIV, that is the cause of AIDS, binds specifically to CD4 surface proteins. The virus uses these proteins to gain entry into helper T cells before it destroys them. Clinically, the progress of an HIV infection and the response to AIDS treatments is monitored by counting the number of cells displaying CD4. Drag the virus to the CD molecule that will allow it to enter its target cell. Every cell in our body has a unique set of proteins on its surface that mark a cell as belonging to self, in other words, one's own body. One major group of these self-antigens is called the MHC proteins. These proteins cradle antigens for presentation to T cells. Remember from Topic 4 that T cells do not recognize free antigens, but instead recognize antigens combined with an MHC protein. Click the cell to see its MHC proteins. MHC stands for Major Histocompatibility Complex. Histocompatibility simply refers to tissue compatibility. MHC proteins determine whether a transplanted tissue or organ will be accepted as self or will be rejected following transplantation. Before any donor organ is transplanted into a recipient, the donor and recipient's MHC proteins are matched as closely as possible. Matching is difficult because there are millions of different combinations of MHC that could be expressed in an individual. This makes it unlikely that any two people will have exactly the same set of MHC proteins unless they are identical twins. Click this potential donor cell to see if its MHCs match the recipient's MHCs. The closer the match, the less likely that the donor organ will be rejected. Click the donor cell again to try to get a better match. In Topic 1, you learn that cellular immunity is directed against intracellular pathogens. Cytotoxic T cells are activated CD8 cells that attack our own cells if they have become infected by an intracellular pathogen, most often a virus, or if they have become cancerous. As cytotoxic T cells circulate through the body, they crawl over the surfaces of all our cells, examining the self-antigens on their surface. How can a T cell know there is something wrong inside a cell by examining the surface of that cell? The answer is Class I MHC proteins, which continuously bring bits and pieces of endogenous proteins, that is, proteins from inside the cell, to the surface for inspection. Click the normal cell to see Class I MHC bringing samples of its proteins to the surface. Normal cell proteins are continuously degraded in the cytoplasm. Special transporters bring these protein fragments into the endoplasmic reticulum, where they are loaded onto newly synthesized Class I MHC proteins. Click the normal cell protein to see loading of Class I MHC. Click the loaded Class I MHC protein to transport it to the surface of the cell. Keep in mind that the process you just saw happens continuously. It is a way of randomly sampling proteins in the cytoplasm and displaying their fragments on the cell's surface. Also remember that during their education in the thymus, T cells have been taught to ignore self-antigens on MHC. Click the antigenic peptide to see the cytotoxic T cell ignore this self-antigen. Drag the virus to the cell below it to infect that cell. Click the viral protein to see it loaded onto Class I MHC and transported to the cell surface. Click the antigenic peptide to see the cytotoxic T cell recognize this foreign antigen. Only a few kinds of cells have Class II MHC proteins. These are the antigen-presenting cells, dendritic cells, macrophages, and B cells. These cells are responsible for communicating with CD4 cells, which are destined to become, or have already become, helper T cells. The antigens that are presented on class II MHC proteins are exogenous antigens, that is, they originate from outside the cell. Let's follow an exogenous antigen on its way to being displayed on a class II MHC protein. Click the dendritic cell to begin this process. Exogenous antigens originate outside the antigen-presenting cell, usually as pathogens such as bacteria. Drag the bacterium onto the dendritic cell to phagocytose it. The next step of the process is the same as for macrophages discussed in Topic 3. The phagosome fuses with the lysosome, and the dead bacteria are digested into small fragments in the phagolysosome. Click the phagosome to fuse it with the lysosome. Class II MHCs are produced in the rough endoplasmic reticulum and are exported to the plasma membrane. On the way, they fuse with the phagolysosome, allowing the fragmented exogenous antigens to be loaded onto the Class II MHC. Click the Class II MHC to see it being loaded and exported to the cell surface. Presenting antigen on Class II MHC proteins has two main outcomes. First, dendritic cells and macrophages present antigen to CD4 cells, triggering them to become helper T cells. Second, B cells and macrophages present antigen to already activated helper T cells to request further activation that will enhance their own effectiveness. Click the bacterial antigen to repeat this animation. Class I and Class II MHC proteins are structurally distinct molecules, but both serve the function of displaying antigens on the surface of a cell. Let's see what you can remember about the differences between MHC1 and MHC2. Use these two choices to fill in the first row of the table. Well done! Now see if you can remember what cell recognizes which class of MHC. That's correct. Remember that normally occurring proteins are displayed on the surfaces of all cells. However, MHC1 and MHC2 can also display foreign antigens. Where do these foreign antigens come from for each class of MHC? Good job! Now imagine that the cell displaying the foreign antigen is saying something to the T cell. What is its message? There is one exception to our generalized messages here, which occurs during activation of CD8 cells. We will discuss this exception on the next page. The activation of T cells almost always begins with a dendritic cell. Dendritic cells capture extracellular antigens directly by phagocytosis and can also capture intracellular antigens from other cells in one of two ways. First, by phagocytizing bits of dead cells, and second, by coming into contact with abnormal cells and receiving antigenic peptides through gap junctions. Because dendritic cells can express high levels of both MHC1 and MHC2, they are primarily responsible for activating both CD4 and CD8 cells. Click the cell debris to phagocytize foreign antigens. When a dendritic cell encounters foreign antigen, the innate immune system signals the dendritic cell to migrate to secondary lymphoid organs. There, it will present its antigens and activate T cells. As it migrates, the dendritic cell matures, expressing large numbers of MHC class I and II proteins, as well as the co-stimulatory molecules that T cells will require to become activated. Click the dendritic cell to start its migration to a lymph node. Earlier, we said that class I MHC proteins present endogenous antigens from the cytosol of a cell. If they are foreign antigens, then this sends the message, "Kill me." However, dendritic cells are the exception to this rule. They have the unique ability to capture exogenous antigens and present them on both class I and class II MHC. Class I MHC on dendritic cells acts as an activation signal for CD8 cells, not as a signal to kill the dendritic cell. In effect, the dendritic cells are saying, "Don't shoot me, I'm just the messenger." Click the dendritic cell to display the exogenous antigen on class I and II MHC proteins. When dendritic cells enter a lymph node, they migrate to the T cell region in the deep cortex. T cells whose T cell receptors match the antigen presented on the dendritic cell will bind to the dendritic cell. Click the dendritic cell to bind T cells to it. However, forming the T cell receptor MHC complex is not enough by itself to activate a T cell. Activation of a T cell requires a two-step process. First, the binding of T cell receptors to MHC proteins bearing antigens. And second, the binding of other co-stimulatory molecules on the antigen-presenting cell. This two-step process prevents inappropriate activation of the T cell. This is like driving a car. The car won't go unless you both start the engine and put it in gear. Click a T cell to add the co-stimulatory molecules. Once the T cells are activated, the processes of clonal expansion and differentiation occur as described in Topic 4. These processes take place in the presence of the cytokines we have discussed, including interleukin-2, which enables clonal expansion. Click a T cell to generate effector cells. While we have shown CD4 and CD8 cells being activated simultaneously, this is not necessarily the case. Each of these T cell types has different effector functions, which we will describe in detail shortly. Click an effector cell to repeat the animation. As we have seen, activation of a T cell is a two-step process requiring both the specific MHC antigen complex and co-stimulatory molecules. Let's compare the response of T cells to antigen presentation with and without co-stimulatory molecules. Drag the T cell to the dendritic cell with a co-stimulatory molecule to see if it becomes activated.
All right, here we go.
Antigen-presenting cells only express co-stimulatory molecules when they have been signaled by the innate defense mechanisms that infection or inflammation is present. When there is no infection, the antigens on the MHC proteins are likely to be self-antigens. Activating T cells in response to self-antigens would be a bad idea. This is why most self-reactive T cells are eliminated during their extensive education in the thymus. However, as discussed in Topic 4, some self-reactive T cells do survive the education process. When these cells react with self-antigen in the absence of co-stimulatory molecules, they are permanently inactivated, a process called anergy. Drag the T cell to the dendritic cell without co-stimulatory molecules.
I don't like this job. I quit.
Even if at some point in the future this anergic cell encounters a dendritic cell with all the correct MHC antigen and co-stimulatory molecules, it will remain inactive. Drag the anergic T cell to the dendritic cell with co-stimulatory molecules to see if it becomes activated.
No, I told you. I quit.
Activated CD8 cells become effector cells, called cytotoxic T cells. Like the foot soldiers of the castle patrolling for enemies, cytotoxic T cells roam the body, searching for a body cell displaying the MHC antigen pair that they recognize. When they find this identifier, they kill the cell displaying it by triggering the process called apoptosis, which we have briefly discussed. Now let's look at this process in more detail. Drag the T cell to the virus-infected cell. Because this is an activated differentiated effector cell, no co-stimulation is required before the cytotoxic T cell delivers its lethal hit. There are two ways a cytotoxic T cell can signal the infected cell to self-destruct. The first way involves the release of pore-forming molecules, called perforins, and hydrolytic enzymes called granzymes. Both perforins and granzymes are contained in granules released by cytotoxic T cells. When a T cell recognizes its target cell, it mobilizes its granules to the site of attachment and releases them into the narrow gap between the two cells. Click the cytotoxic T cell to release its granules. The perforin molecules that have been released elongate and insert themselves into the membrane of the target cell where they group together to form a pore. Click the space between the cells to create pores. The pores formed by the perforins are similar in size and shape to those of the membrane attack complex formed by complement. Like the membrane attack complex, they allow movement of molecules in and out of the cell. Among the molecules that enter the target cell through the pores are the granzymes released by the cytotoxic T cell. Click a pore to allow granzymes to enter the target cell. Once inside the cell, the granzymes activate certain other enzymes, beginning the pre-programmed process of self-destruction that is apoptosis. Click a granzyme to trigger apoptosis. This first way of triggering apoptosis, using perforins and granzymes, is assisted in many cells by a second mechanism. This mechanism uses a special type of apoptosis-inducing receptor on the surface of these cells to which the cytotoxic T cell can bind, triggering apoptosis. Cytotoxic T cells and the natural killer cells of innate immunity share these two killing mechanisms. These cells also share the job of immune surveillance, patrolling the body for abnormal cells. They differ in that cytotoxic T cells look for foreign antigens on MHC, while NK cells look for the absence of MHCs. NK cells are thus able to eliminate abnormal cells that cytotoxic T cells cannot detect. Despite what you might think, cytotoxic T cells are not the most important T cells. In fact, helper T cells, which outnumber cytotoxic T cells by 2 to 1, are more important to the immune system, since without them, there is essentially no adaptive immune response. Helper T cells are almost always required for activation of both B cells and cytotoxic T cells. As we've seen in Topic 5, helper T cells are critical for the activation of B cells and the humoral immune response to most antigens. To briefly recap, a B cell that has captured its specific antigen presents the antigen on class II MHC protein to an already activated helper T cell, which then provides the co-stimulatory signals necessary for full B cell activation. Click the B cell to activate it. The B cell then undergoes clonal expansion and differentiation into antibody-secreting plasma cells and memory cells. Click the helper T cell to see how it helps activate cytotoxic T cells. CD4 cells, which become helper T cells, are more easily activated than CD8 cells, which become highly destructive cytotoxic T cells. A mature dendritic cell expresses enough co-stimulatory molecules that it can activate CD4 cells, but not usually enough to activate CD8 cells. Click the CD8 cell to see if it will be activated by the dendritic cell. No activation occurred because the CD8 cell's co-stimulatory molecule has no counterpart on the dendritic cell to which it can bind. If a helper T cell recognizes antigen presented on class II MHC proteins by this dendritic cell, then it can help activate the CD8 cell. Click the helper T cell to bind it to the dendritic cell. The helper T cell can help in two ways. First, it can stimulate the dendritic cell to express additional co-stimulatory molecules. Click the helper T cell to signal the dendritic cell, telling it to add co-stimulatory molecules. Second, the helper T cell can secrete cytokines, including interleukin-2, that help activate the CD8 cell. Click the helper T cell to release interleukin-2. As you have seen, helper T cells activate both arms of the adaptive immune response. The importance of helper T cells becomes abundantly clear in situations where they are deficient, such as in acquired immune deficiency syndrome, AIDS, a disease in which CD4 cells are selectively destroyed. In this disease, both cellular and humoral immunity are compromised. In effect, there are no officers directing the troops. CD4 cell can differentiate into one of two broadly defined types of helper T cells, depending on the type of threat perceived by the innate immune system. These two classes of cells, called Th1 and Th2, help the immune system fight different types of pathogens. Click the dendritic cell to begin exploring Th1 cells. Th1 development is triggered by the presence of hard-to-kill bacteria in macrophages and by viral infections. Click the bacterium in the macrophage to see which cytokines promote differentiation into Th1 cells. Notice that the innate defenses trigger the release of interleukin-12 as they sound the alarm, and thereby determine the type of response provided by the adaptive immune system. Click the Th1 cell to see how it helps. Th1 cells secrete gamma interferon, which boosts the ability of macrophages to kill engulfed bacteria. Gamma interferon also promotes cell-mediated immunity by cytotoxic T cells. Dealing with the intracellular pathogens that trigger Th1 formation is the last step in this negative feedback loop. Click the dendritic cell to begin exploring Th2 cells. Th2 development is triggered by extracellular pathogens. Click an extracellular bacterium to see which cytokines promote the differentiation into Th2 cells. Notice that the innate defenses trigger the release of interleukin-4 as they sound the alarm, and thereby determine the type of response provided by the adaptive immune system. Click the Th2 cell to see how it helps. Th2 cells secrete interleukins 4 and 5, which promote B cell activation and development and the release of certain classes of antibodies. Click the B cell to see the consequences of the Th2 response. Click either the Th1 or Th2 cell to repeat its portion of the animation. Click either the Th1 or Th2 cell to repeat its portion of the animation. Click either the Th1 or Th2 cell to repeat its portion of the animation. As we've seen, the helper T cells help activate effector cells of the immune system. This activation process contains a number of safety checks whose purpose is to keep the immune system from being activated when it is not required. We will now consider our last class of T cells, the regulatory T cells, whose main function seems to be to suppress the activity of self-reactive cells and prevent autoimmune diseases like the 4 examples illustrated here. Click any disease to learn more about preventing autoimmunity. Unlike the mechanisms of self-tolerance we have seen before that involve clonal deletion, regulatory T cells actively suppress other T cells. How might they do this? Let's briefly review the generation of effector T cells and consider how regulatory T cells can interfere. Click the thymus. As we've seen, the creation of effector T cells begins with the recognition of antigens presented by an antigen-presenting cell together with the appropriate co-stimulation. This leads to proliferation and differentiation of the naive T cell into effector T cells, which then carry out various functions. Regulatory T cells are also effector T cells. They are derived from the same line of CD4 expressing cells as helper T cells in the thymus. There are at least 3 major kinds of regulatory T cells. While we do not fully understand their properties and how they work, let's explore ways in which they are believed to work. Click the thymus to continue. As we have seen throughout the immune system, there are really only 2 ways in which immune cells work, either by direct cell-to-cell contact or by the release of chemical messengers such as cytokines. Regulatory T cells are thought to do both. Click a regulatory T cell to see inhibition via cell-to-cell contact. Click another regulatory T cell to see inhibition via cytokine release. The properties of regulatory T cells and their potential use in treating autoimmune diseases and preventing transplant rejection are areas of intense research. We have now completed our discussion of cellular immunity. Through our study of the immune system, you have undoubtedly gained an appreciation for the breadth and depth of the body's defense systems, beginning with the surface barriers, chemicals and cells of innate defenses, and the humoral and cellular arms of adaptive defenses. Just like the defense system of a castle with its soldiers who defend it, all of the components of the immune system act together to defend the body against those elements that would do it harm. Here's a summary of what we've covered. Cytokines are small peptides that mediate interactions between immune system cells. CD8 cells become cytotoxic T cells. CD4 cells become helper T cells, Th1 or Th2, or regulatory T cells. Class I MHC proteins, found on all cells, present endogenous antigens to CD8 cells. Class II MHC proteins, found on antigen-presenting cells, present exogenous antigens to CD4 cells. T-cell activation requires antigen presentation by an antigen-presenting cell along with the appropriate co-stimulatory molecules. Cytotoxic T cells induce apoptosis of target cells by releasing perforins and granzymes. Helper T cells are required for activation of most B cells and CD8 cells and release cytokines to direct the immune response. Regulatory T cells act to suppress self-reactive T cells, preventing autoimmune diseases. To test your knowledge, click the quiz button to go to the self-quiz. To access cross-references for this topic in your Benjamin Cummings textbook, click here. To test your knowledge, click the quiz button to go to the self-quiz.